students, do you know how plants burst their energy in the form of photorespiration, and which is the pathway involved during this process? It is a C2 pathway which is involved during the process of photorespiration. In this video, we will discuss in details how the C2 pathway is taking place in the plant. So before that, we have to understand what is photosynthesis and what is photorespiration. Photosynthesis, it is a process by which the carbon dioxide is taken by the plant and it gives oxygen. And photorespiration is alternate to it. It is opposite to it. That means the plant is taking the oxygen and giving carbon dioxide. It is utilizing the energy. It is not giving any energy. It is utilizing the energy. So let us see how this pathway is taking place, how the photorespiration is taking place in the plant. The process of photorespiration is in the C2 pathway. The C2 pathway is taking place in chloroplast, peroxisome and mitochondria. These are the three organelles which are involved during the process of photorespiration. And it starts with RuBP. It is ribulose 1,5-bisphosphate. Two molecules at first in the chloroplast. There is two molecules of ribulose 1,5-bisphosphate. This ribulose 1,5-bisphosphate, it is being converted into 2-phosphoglycolate and 3-phosphoglycerate. This 3-phosphoglycerate which is formed, it is a 3-carbon compound and it is being utilized in the Calvin cycle. The next is a 2-carbon compound which is present, which is known as 2-phosphoglycolate. This is involved during the process of C2 cycle or during the process of photorespiration. So during this process, there is an enzyme which is involved, which is known as RuBisCO. This RuBisCO, it is having both oxygenase as well as carboxylase activity. In presence of oxygen, this RuBisCO is working as a oxygenase, and during the process, during the formation, during the C2 cycle or during photorespiration, the oxygen is being utilized here. Here two molecules of oxygen is being utilized here in presence of the enzyme RuBisCO, and it is converted into 2-phosphoglycolate. This 2-phosphoglycolate, now it is being converted into glycolate. Here there is removal of phosphate over here. So for removal of the phosphate here, H2O is being involved. So here 2H2O is involved for removal of this 2-phosphate. Okay. So as here there is removal of phosphate, there is a enzyme which needs to be present here. So the enzyme which is present here is phosphoglycolate phosphatase. Okay, it is phosphoglycolate phosphatase. I will write here. It is the phosphoglycolate phosphatase. It is the enzyme which is helping for conversion of 2-phosphoglycolate into glycolate. And here there is utilization of H2O for removal of 2-phosphate. Now this glycolate which is present, it is being transferred into other organelles which is known as peroxisome. Okay. Now this 2-glycolate is being converted into 2-glyoxylate. Okay. So the conversion of 2-glycolate into 2-glyoxylate, during the process there is removal of water from it. So here there is 2O2 is being utilized again over here and it is forming 2H2O2. And in the peroxisome there is an enzyme which is known as catalase. It is breaking this 2H2O2 into 2 molecules of H2O. Okay, and during this process there is involvement of another one enzyme which is known as, here I am writing, it is known as glycolate oxidase. It is glycolate oxidase. Okay, so now 2-glycolate. Now the 2-glycolate is converted into 2-glycine. This glycine which is present, it is having a NH3 group with it. Okay, here there is a NH3 group in it. So here also, as it is being converted into a amino acid which is known as glycine, from where this NH3 is coming. So this NH3 is coming from another one amino acid which is known as glutamate. This glutamate is giving the NH3 to it, and this glutamate is converted into alpha ketoglutarate. Here 2 molecules of glutamate are utilized and it is being converted into 2 molecules of alpha ketoglutarate. And here also there is involvement of one enzyme. The enzyme which is involved here is glutamate. It is glutamate glyoxylate aminotransferase. It is aminotransferase, as it is transferring the amino acid. Okay, so here during the process of when the glyoxylate is converted into glycine, here there is NH3 group which is present. Here the enzyme which is working is glutamate glyoxylate aminotransferase, because one amino group is being transferred here. And from where does this amino group is coming? It is coming from 2-glutamate. 2-glutamate is giving the amino acid and it is being converted into alpha ketoglutarate. Okay. Now this glycine which is present, it is being transferred into mitochondria. Here we can see it is being transferred into mitochondria and converted into serine. Okay, this glycine which was present, it was a 2-carbon containing compound. It was a 2-carbon containing compound, but it is having 2 molecules of 2-carbon containing compound. So here what happens? Here there is 4-carbon containing compound, but here the serine which is present, it is a 3-carbon containing compound. So here there is 1 extra carbon. So this 1 extra carbon is being removed over here by the utilization of water. Okay, so here the enzyme which is involved is glycine D-carboxylase. Okay, apart from it, there is conversion of NAD plus into NADH. Okay, and the NH4 group is being released. So here the other enzymes which are working is serine methyltransferase. Okay. So now the serine which is present now in the mitochondria, it is being transferred into the peroxisome. Here again it is being transferred into the peroxisome. Here we can see the serine over here. So this serine which is present here, it is a 3-carbon containing compound. Now this serine is converted into hydroxypyruvate. Okay, so here also there is no need of any NH3 group. Here we can see here NH3 is present. So this NH3 is given to alpha ketoglutarate. So this alpha ketoglutarate will take the NH3 with it and it will be converted into glutamate. From here, one NH3 group is being given to alpha ketoglutarate and it is being converted into glutamate. Okay. So now next what happens? Here there is involvement of another one enzyme which is known as aminotransferase. Okay, now the hydroxypyruvate which is present. Hydroxypyruvate will be converted into glycerate. During this time NADH is being converted into NADH. The NADH which is present over here, it was from this part. When the glycine was converted into serine, there was release of one NADH. This NADH being utilized over here. The NADH is converted into NAD plus when the hydroxypyruvate was converted into glycerate. Okay. So next, now this glycerate which is present, it will be transferred into the chloroplast. Okay, so after this glycerate is being transferred into chloroplast, it will be converted into 3-phosphoglycerate. That means here 3-phosphate is being added over here. So here there is utilization of ATP. Okay, and there is involvement of another enzyme which is known as kinase for converting this glycerate into 3-phosphoglycerate. Okay, now this 3-phosphoglycerate which was produced in this part, like when this ribulose 1,5-bisphosphate in presence of ribose, it was converted into 2-phosphoglycolate and 3-phosphoglycerate. This 3-phosphoglycerate and this 3-phosphoglycerate which has been bought by the glycerate, it undergoes into Calvin cycle. Okay, so now here the alpha ketoglutarate which was present, from here one alpha ketoglutarate took the NH3 and it converted into glutamate. Now there was another one alpha ketoglutarate. This alpha ketoglutarate will be transferred into the chloroplast. Okay, this alpha ketoglutarate will be transferred into the chloroplast and it will help in the formation of glutamate. Who will help in the formation of the glutamate? The NH4 plus which was formed during the conversion of glycine to serine. This NH4 plus, it will help in converting this alpha ketoglutarate NH4 plus which is present. It will help in transferring this alpha ketoglutarate into glutamate. Okay, it will help and it will take the NH3 with it. Okay, now what happens? Now this glutamate which is produced, again it will utilize in the peroxisome. This glutamate which is present, it will be again present in this peroxisome and it will give the NH3 group to this and it will form the alpha ketoglutarate. In this way, the C2 cycle is taking place, or we can tell that the Calvin cycle is taking place. So in the Calvin cycle, there is involvement of 3 cell organelles, like it can be chloroplast, it can be peroxisome and mitochondria. So let us have a recap again. So it is taking place in presence of the enzyme RuBisCO. So here the ribulose 1,5-bisphosphate is working and this RuBisCO is working as a oxygenase. Oxygen is being utilized over here. Next is it is being converted into 2-phosphoglycolate and 3-phosphoglycerate. This 3-phosphoglycerate is going into the Calvin cycle and 2-phosphoglycolate is going into the C2 cycle. During this time there is removal of phosphate group. So during this removal there is involvement of 2H2O and phosphate is being removed. Now it is converted into glycolate. Glycolate it is transferred into the peroxisome. In the peroxisome it is using the oxygen and converted into water and transferred into glyoxylate. Glyoxylate is converted into glycine which is having a NH3 group with it, and this NH3 group is provided by this glutamate. By transferring the NH3 group, it is forming the alpha ketoglutarate. Now this glycine is being transferred into mitochondria, from which forms the serine by the release of carbon dioxide and by production of NAD plus into NADH, and NH4 plus is being removed. Now the serine, it goes into the peroxisome. From the peroxisome it is converted into hydroxypyruvate. During this time the NH3 group which is present, it is being taken by the alpha ketoglutarate and it is being converted into glutamate. Now this hydroxypyruvate which is formed, it reduces the NADH into NAD plus and convert into the glycerate. Glycerate is being transferred into chloroplast. From the chloroplast there is utilization of 3-ATP UTP and it forms a 3-phosphoglycerate, and this 3-phosphoglycerate is taking part in the Calvin cycle. This is how the C2 pathway is working during the photorespiration. I hope you have understood this lecture very clearly. If you have not understood, if you are facing any doubt in this video, please do ask me in the comment section. Thank you everyone. Thank you for watching this video till the end. Thank you.